In this lecture, we'll be talking about cell function and structure. We'll start off by talking about the discovery of the cell. In 1665, Robert Hooke and within this book, he describes what he viewed under the microscope as box-like structures. He later coined the term as a cell in reference to the box-like structures that he was seeing under the microscope. In the 1670s, then Leeuwenhoek discovered bacteria and protozoa. And as the time went on, more advances in science led to the increased development of the lenses, microscope construction, and staining techniques. These advances allowed scientists to see more of the components within a cell. Now, there are several different types of microscopes that can be used and are used. Some of these include the light microscope, the electron microscope, and the scan and probe microscope. We will focus more on the light microscope because it is one that is known by more people. It is used more often. Here I have examples of the electron microscope and the scan and probe microscope, but these are here more to show that there are several different types of microscopes. Now, the light microscope is used most often in academic settings as well as in medical laboratories, and they have lens objectives that can magnify 40 times, 100 times, 400 times, and 1,000 times. These are the usual objective lenses that can be found on the light microscope. And the 1,000 magnification objective lens is also referred to as the oil immersion lens because you actually need a drop of oil on your slide when you're using this lens. What you do is you take your slide that has your cells or your microorganisms, place it onto the microscope stage. You take a drop of oil and put it on that slide, and then you're able to view it with the 1,000 magnification objective lens. Next, we're talking about the endosymbiont theory. It's also referred to as endosymbiosis. So if you hear endosymbiont or endosymbiosis, it's the same thing. Now, endosymbiosis points to the reason why mitochondria, the thought behind why mitochondria and chloroplasts may contain their own DNA and ribosomes. So scientists noticed that bacteria, mitochondria, and chloroplasts are all of similar size. And it was also discovered that bacteria contain DNA just as chloroplasts and mitochondria do. So it is believed that host cells and bacteria formed an endosymbiont relationship, and then when the cell ingested both the aerobic and autotrophic bacteria, it didn't destroy them. Instead, a mutualistic relationship or beneficial relationship was formed there. And through many years of evolution, the ingested bacteria became more specialized and with the functions that it was able to do, and with the aerobic bacteria becoming what we're referring to it as mitochondria, and the autotrophic becoming the chloroplast. Now here is an example or a simplified way or version of the endosymbiont theory. You have two independent bacteria. One engulfs the other, and then the one that is engulfed ends up living inside of the other. Both end up benefiting from the arrangement, and as subsequent generations are produced, the bacteria is just passed on from generation to generation. So the engulfed bacteria is just used by each generation, is passed on from generation to generation. The cell theory just states that the three main topics or three main points, and these points are that the cell is the basic unit of life. All cells are composed of one or more cells, and then all new cells or pre-existing cells arise from already existing cells. Now we go on to discuss prokaryotic versus eukaryotic cells. Now, these images show, the one on the right is an image of the eukaryotic cell. It shows the different organelles that are within them, and these organelles help with the functioning of the cell. On the left, we have a prokaryotic cell, and within it, you can see internal structures, but we don't consider these structures as organelles. These are just structures within the cell that help it function. Prokaryotic cells are usually smaller than eukaryotic cells, and this provides advantages and disadvantages on both ends. With the eukaryotic cell being bigger, it presents some disadvantages. But with them having organelles that can do perform several different functions, is actually an advantage for them. Eukaryotic cells are usually more complex than prokaryotic cells are, so they're sometimes able to perform more functions. Prokaryotic cells, pro means before, and kary means nucleus. So this means that prokaryotic cells are cells that do not have a nucleus. Prokaryotic means before nucleus, so no nucleus is found in prokaryotic cells. These are single-celled, simple organisms that lack nucleus. The organisms contain DNA that can be found within the central part of the cell, called a nucleoid. And prokaryotic cells are usually broken down or distributed between two domains, bacteria and archaea. Now, all prokaryotic cells have some type of chromosomal DNA localized within the nucleoid. All prokaryotic cells also have ribosomes, the cell membrane, and the cell wall. Now, as I previously stated, all prokaryotic cells do have the cell membrane, cell wall, ribosomes, and the nucleoid that contains the DNA. If you look at the picture here, you can see what these things may look like within a prokaryotic cell. Ribosomes are the very tiny circles that is seen in this image. The nucleoid region is kind of big because it contains, big for a prokaryotic cell because it contains the DNA of that cell. Some structures that are found in prokaryotic cells, but not all of them, includes the cytoskeleton, pili, flagella, cilia, and fimbriae. Within the cell wall that all prokaryotic cells contain, most will have a peptidoglycan cell wall, and some, the ones that do have a capsule, will usually have a polysaccharide capsule. And the cell wall surrounds the cell membrane and acts like an extra layer of protection, helping to maintain the shape and prevent dehydration of the cell. The capsule, if one is present, permits the cell to attach to surfaces within its environment. And some bacteria that contain pili, flagella, or fimbriae use these things mostly for locomotion. Pili can be used for several different things, but is mostly used for exchange of genetic material, whereas fimbriae is used to attach to a host cell. If a cytoskeleton is present, it is more like a protein framework that is involved in the cell division and helps also with the shape of the cell. And the ribosomes that are found in all prokaryotic cell structures is used for protein synthesis of for the cell. It provides the energy. It provides the proteins that's needed for the prokaryotic cells. Now here we have classic examples of prokaryotic cells. On the right, we have two images of the euglena, and this is a classic example of the flagellated prokaryotic cell. The structure that you can see moving is the actual flagella that's present within the euglena. Now on the left, we have a paramecium, and this is the classic example of the ciliated prokaryote. You can't really see the cilia in the image, but if you the movement of the cell is being caused by the cilia that's present all around the cell, all around the paramecium. So both cilia, and flagella are used for movement of prokaryotic cells. Now we'll talk about the 3 basic prokaryotic cell shapes. These are not the only shapes that prokaryotic cells can be found in, but these are the 3 main ones. And if you look at the first one, it's cocci, and it's usually, it looks like circles or spheres, and these shapes can be in chains or they can be in clusters, but you're still going to be able to see the shape, the circular shape of the individual bacteria. The next is the bacilli, sometimes also referred to as rods, and it does have that rod shape. If you look at the image there, it is a rod shape. And for spirilla, also known as spiralis or spirals, it has a spiral shape to it. Here's just another image of, like a closer image of those same bacterial shapes. So the spherical one is also cocci, is the scientific name for it, but as you can see, it's circles. Rod shape, same thing as bacillus, and it actually has those rod shapes. And then the spiral or spiralis has the spiral shape. Now on to eukaryotic cells. Eu means true, breakdown of the word. Eu means true, kary means nucleus, meaning true nucleus. So eukaryotic cells are those that actually have a true nucleus. They have a membrane-bound nucleus. Animal cells, plant cells, fungi, and protists are all made of eukaryotic cells. Eukaryotic cells will have a membrane-bound nucleus. They can have chromosomes as well as membrane-bound organelles. And some of these membrane-bound organelles include endoplasmic reticulum, the Golgi apparatus, chloroplast, and mitochondria. There are many other organelles that are also found within the eukaryotic cells, but this is just an example of a few of them. So here we have an image of the eukaryotic animal cell, which in eukaryotic cells can be broken down into plant and animal. Some of the organelles are shown here in this image, and this includes things like the lysosomes, mitochondria. You have rough endoplasmic reticulum and smooth endoplasmic reticulum. And of course, you have the nucleus that contains all the genetic information in the chromosomes of the cell. Now here is an example of a eukaryotic plant cell. Now within this cell, it has a lot of the same structures that a eukaryotic animal cell has, but it has a few things that an animal cell would not have. One of those things is the mitochondria. If you look at the image and look at these large green areas, this is a representation of the chloroplast. And the chloroplasts are only found in eukaryotic plant cells. You will not find chloroplasts in an animal cell. This large vacuole is also specific to a eukaryotic plant cell. Although animal cells do have smaller vacuoles that are sometimes referred to as vesicles, the plant cell has a large one, whereas the animal cell has small ones. Now, other cell structures such as the nucleus, cell membrane, Golgi apparatus, ribosomes, mitochondria, this can be found in both plant and animal cells. And in addition to chloroplasts being only found in plant cells, most animal cells do not have a cell wall. The cell wall is only found in the eukaryotic plant cell and not the eukaryotic animal cell. Now here's a breakdown of some of the organelles and structures that are found within plant and animal cells. Now a lysosome is an organelle that contains digestive enzymes, and their main purpose is to digest excess or either worn out organelles, food particles, engulfed viruses or bacteria. Their purpose is to digest or destroy these things. And the lysosomes can be found in animal and plant cells. Now the nucleus, as discussed earlier, is only found in eukaryotic cells. And the nucleus is a membrane-bound organelle that contains the information system or the genetic information for the cell. And a nucleus can be found in both plant and animal cells, but not in prokaryotic cells. Mitochondria is considered the energy factory of the cell. And it's like the cell's energy factories. It's the energy factory of the cell. The mitochondria will manufacture the ATP that fuels the cell's activities. It serves as the site of aerobic cellular respiration, and it can be found in both plant and animal cells. The Golgi apparatus serves as the site of synthesis. It does the packaging of certain molecules inside of a eukaryotic cell. And once again, it can be found in both plant and animal cells. Ribosomes are the protein factories of the cell, and its job is synthesizing and building protein and polypeptides, and it too can be found in both plant and animal cells. Now, the vesicle, as I referred to earlier, is sometimes referred to as vacuole. It's small vacuoles in animal cells. So if someone says a vesicle or vacuole in an animal cell, most likely they're referring to the same thing. But they can be found in plant and animal cells. However, in an plant cell, it's going to be a much larger vacuole than in plants and then in animal cells. Animal cells are small. Plant cells is a large vacuole. And here is an image of chloroplast. And chloroplast is the area where the photosynthesis takes place within a plant cell. And they are responsible for converting energy. They're sac-like organelles, and they actually contain the chlorophyll that the plant uses. And chloroplasts are found only in eukaryotic cells, eukaryotic animals, plant cells, excuse me. Here we just have an image or of a comparison of prokaryotes and eukaryotes. And it's just a brief description. We've already discussed all of this, but this just shows you a comparison of the two, how the prokaryotic cell, it lacks a nucleus, it doesn't have membrane-bound organelles, whereas a eukaryotic cell is more complex, and it has those organelles that have a membrane around them, and it also has more structures inside of it that helps it to perform more complex tasks. And here's another image that just shows a comparison of the prokaryotic cell with the eukaryotic plant cell and the eukaryotic animal cell. It just shows you the differences and the similarities and all of them, and what you can expect to see when you're looking at prokaryotic and eukaryotic cells and what structures should be found in which ones. Considering what we've already talked about, here's some questions that should be able to be answered. Which cell could be considered a eukaryotic cell, and which one should be considered a prokaryotic cell by this image that's shown? If you look at the image on the right, it does not have a nucleus. It does not have a lot of structures inside of it. There are no membrane-bound organelles, but you can see a central section of DNA, or what we can assume is DNA. Looking at the image on the left, we see an area that looks like DNA surrounded by a membrane. We can see what looks like mitochondria, smooth and rough ER, other things that look like the Golgi apparatus. So we can assume that the image on the left is a representation of a eukaryotic cell, while the image on the right is a representation of a prokaryotic cell. And judging by the information that we've covered, all of these questions should be easily answered. Name one structure that is found in plant cells but not in animal cells. One structure that is found is chloroplast. Okay, name one of the three basic shapes of prokaryotic cells. There are three different basic shapes for prokaryotic cells. Those are the cocci or spherical shape, bacilli, bacillus, rods, all that's the same thing, and the spirilla or spiral shape. Okay, name two types of eukaryotic cells. There's only two types of eukaryotic cells, a plant cell and an animal cell. That's it. Then name one type of microscope that can be used to view cells. We only discussed three types specifically. Only one of those are used mainly in academic and medical laboratory settings, and that is the light microscope. But electron microscope can also be used to view cells, as well as the scan and probe microscope and a ton of other microscopes as well. And the last question, the blank is the basic unit of life. That is the cell. The cell is the basic unit of life.